I'm presenting here a seminar, a lecture on structures and evolution of proteins. So I am thankful to Hervé because some of the material which will be covered has already been explained by Hervé. So that's very good because I can shorten a little bit the lecture. The plan of my lecture is the following. First of all, I will discuss what are proteins with, in a shortened version, as I said. Then I will explain some basics about the protein structure. And finally, I will discuss the astrobiological relevance of proteins. So let us start. And of course, we don't need all this part about proteins because they have been already explained by Hervé. Just for me, it is very important here to recall that proteins are based on amino acids, and they have here an alpha carbon. Then they have an amino group and a carboxyl group. So this is the structure of the amino acids. And as it was already recalled, proteins are looking like words. And these words have an alphabet of 20 letters. And the letters are the 20 possible amino acids, which are distinguished by the side groups. So I'm very fast at that point because we have already seen this picture in some other lecture. So of course, for a physicist, what it is important here is that these molecules are polar. So not all, but they sometimes they are charged. So their characteristics from the physical point of view is different. However, many molecules, so many amino acids are interchangeable. So this is an information that it is important to remember. Now, let me skip, however, to the what I'm calling, and it is usually called, the protein paradox. I'm calling it the first protein paradox because there will be another. So proteins have an alphabet of 20 letters, and this is not very much compared with the 26 letters of English. However, the words are extremely large because in English we have words that contain from 1 to 12. Of course, there are bigger words, but these are exceptions. From 1 to 12 letters. Here, proteins contains from 100 to 1,000 amino acids. So they are very, very long words. And if you can check the orthography, the grammatic of such words, the number of possible combinations is incredibly large because let me remember that proteins can be any sequence of amino acids. And so if you don't consider the longest proteins, but if you imagine that we have proteins that in the average consist of 200 amino acids, then the number of combinations is very huge because if you count is 20 to 200. So in the basis of 10, this means 10 to 260. So now let's imagine that we have a huge laboratory. So huge means the whole earth. The earth as a radius of as a surface of about 5 times 10 to 8 kilometers square. And let us suppose that we can take a 10 kilometer thick layer of these surfaces and fill it with proteins, and all proteins inside this volume will be different. So in that way, we will be able to fit only about 10 to 34 chains. And now let's assume that we have all the time of the world, and this is literally. So all the 4.5 billions of years history. So even if your laboratory is very, very fast, you have a very, very complicated and fast equipment which is able to work in the time of 10 to minus 11 seconds, which is comparable with the time of molecular collision, then the number of combinations of different amino acids in proteins that you can test is 10 to 34 times 10 to 28, which means 10 to 62, which is very, very far from the number of all possibilities, which is, I remember, 10 to 260. So how could evolution have progressed when there was no opportunity to take the best choice? Because it is clear that even the Earth, even the entire universe could have explored just a few combinations with respect to the entire spectrum of combinations. So this doesn't look very encouraging for astrobiology, but actually it is not so bad, because in practice I think that the nature chose a little bit at random, and there are evidences about that. About this I will come back later. Now there are a few common factors in proteins which I don't want to explore in detail, but one is important. You can see here the way in which amino acids are assembled inside proteins. So you can see here we have two amino acids. R is the special group, as Hervé told before. They are connected with carbon atoms, which is called the alpha carbon, and they connect themselves by eliminating here the oxygen atom together with the hydrogen atom, with that hydrogen atom in the second amino acids. And in this way, they form what it is called a peptide bond. And as you see, a peptide bond is a bond between a nitrogen atom and a carbon atom. So this is repeated twice. So for this reason, it's called dipeptide. But what it is important here is that we have here always at the left side nitrogen atom, and here at the right side you have a carbon atom. If you add other amino acids in that way, you will always have here in the left a nitrogen atom, and on the right a carbon atom. Of course, sometimes it can be that the carbon atom in the left and the nitrogen on the right, but this is always the same. You have an end with a carbon and one end with a nitrogen atoms. So here you can see that this part with the nitrogen atom is called an N terminus, and this part with this end with the carbon atom is called a C terminus. And always when proteins are translated inside the living organism from the messenger RNA, they always start from a free amine group. So this containing this NH2 molecule, and they are always terminated by a free carboxyl group. This C O2H part, which is actually acid. So this is why they are called amino acids. Now, there are also a few other facts that should be taken into account. That the proteins have functions. One of the function of the protein is that they are working for scaffolding. So that the so-called structural proteins, which from the astrobiological point of view are rather boring because they're very, very regular, and they present a periodic structure which is needed to build various structures inside cells and inside living organisms. More interesting are the so-called functional proteins, which are usually globular proteins. So globule is very compact, so they are rather compact, and they are functional in the sense that act as enzymes. So we have an enzyme here, as you can see, and this is the schematic representation of our protein. And here there is a substrate molecule to which this protein will be bound. So here I present a lock and key model of the protein, which is not the only one model. So this predicts that this part of the proteins fit exactly or almost exactly with the substrate molecules. And the idea is that these functional proteins have active sites, which are these sites where the substrate molecules are binding, and these structures, just to recognize which molecules should be bonded, is folded in a particular way. And so this is why this is one of the reasons why proteins are folded. And once the proteins is binded with the substrate molecule, it starts a reaction. It contains a part inside this fold which is not so rigid like the fold, but it is rather flexible. And this part starts some reaction, and the result is that the molecule, the substrate molecule, is splitted in products that are smaller. Now, proteins have a very, very complex objects, and for this reason they look differently according to the graphical representation. For instance, here in the bottom, we just show a lattice representation, which is used in general to make computer simulation of polymers, and in particular to find how proteins can fold. And here we have other structures, the wireframe diagram, in which you can see how the single atoms of the protein are bonded together. Here you have the same structure, but in which the atoms are surrounded by spheres, and at the end we have the so-called backbone and ribbon diagram. If you want to see the shape of the protein, this space filling diagram is the best. If you want to study it from the chemical point of view of a protein, and to see how it acts as an enzyme, where are the active sites, then the wireframe diagram is better. If you want to see how the protein is folded, then the backbone and ribbon diagram is best. Now, we should also take into account that, of course, this backbone, so this purple chain, and this is held together by strong covalent bonds. So these are the strongest chemical bonds in living systems. Now, however, what it is here important is that this is not like a crystal in which the molecules can just move around some equilibrium point. No, here the molecules can move very, very fastly and have a lot of degrees of freedom. And what it is more important is that different parts of the biomolecules of the protein can be connected by various weak bonds, like hydrogen bonds, disulfide bridges, or even weaker van der Waals forces. And now what it is important is that these weak bonds can be easily broken by thermal fluctuations, and then they can also be reconstructed. But they give to the proteins their incredible amount, an incredible amount of slightly different configurations, which are held together. And from the energetical point of view, they are more or less stable. And this is why the protein can fold in a number of different configurations. And the energy landscape of such folding, so how the energy of different configuration is distributed is shown here at the bottom. So you see that you have a landscape which is very, very complicated. So proteins are complex systems, and in this top and valleys, the valleys are the minima, and there are really, really many. In comparison, you see the situation of the atoms in a solid, for instance, in the case of crystal, that they have just a single minima at some particular point. So, I'm now ready for the second protein paradox. We have seen already that there is a paradox with reproducing all the astonishing number of proteins. The second protein paradox is the following. A protein with a given primary sequence, I didn't say what is a primary sequence, but it is the sequence of amino acids along the chain, can fold in a very large number of slightly different conformational substates. And so from the point of view of statistics, each building block, so each amino acid inside the protein can, on average, assume from 2 to 3 different configurations. And since, as I said, approximately we can imagine that in average there are 200 amino acids in proteins, then the number of such states, the number of different possibilities of configuration is enormous, because once again is going from 2 to 3 to 200 to the power of 200, and all the states have an approximately equal energy. So if you remember that proteins fold in an extremely short time, which is of the order of 5 milliseconds, so they get a particular shape, which is always the same. So how the proteins can explore all this wealth of possible states in a so short time. And so there are a few proposals about this solution, but actually this problem is still unsolved. The second protein is the so-called Levinthal paradox. And the possible solution of the paradox is to suppose that many configurations, almost all the configurations, have more or less the same energy. So we see here that we have a landscape which is inside the funnel, at the border of the funnel, almost flat, so not very complicated. But the funnel structure is given by this N, which is the native structure, so the folded configurations, which has a real low energy state. And so at the end, the protein chooses exactly this lower energy state, which is very favored. Another possibilities, the hypothesis of Trifonov that I will explain later. And this assumes that proteins contain shorter subunits of about 25 or 30 amino acids that fold independently. So for these subunits, the folding, the possible configurations are much less, and so this would explain the paradox. Now let's go to protein structures. I will just do a very brief summary, and essentially this is just what matters. We have a primary structure, which is the sequence of amino acids, a secondary structure, which is a short sequence of amino acids from about 10 or even less amino acids, and then the tertiary structure that puts together many secondary structures. But let me notice that even the tertiary structure is very, very small, because the size is about of this small volume is about 4 nanometers squared. So, so the primary structure, as I said, is the sequence of amino acids in a protein. And what is I'm recalling what I said before, that let us note that the primary structure has a high degree of randomness, despite the fact that life has now almost 4 billion of years. So this is, I believe, connected with the fact that it is difficult to explore all the possibilities. So it is difficult to find the best protein in the world. So simply at the beginning there was an almost random choice, the first choice that has worked out. Now the secondary structure. Here, this is a little bit more difficult to explain, but you can see here the structure of the chain that is forming the proteins. So we have these amide groups that are connected together by these side groups, and actually, from the point of view of geometry, from the three-dimensional point of view, these two parts of the proteins are in two different planes. Those connected by this alpha carbon, and these planes geometrically, their orientation can be described by two angles. One is the angle psi, and one is the angle phi. There is another angle, but this is not so important, so I will skip it. And depending on the values of phi and psi, and if they always the same values are repeated, then what happens is that regular structures are forming along the chain. For example, repeating values of phi is about minus 57 degrees and psi about minus 47 degrees give a right-handed helical fold that I denoted here in this way by a spring. There are also other structures. One are the so-called beta sheets, which are corresponding to other repeating values of triangles, phi and psi, and many others. For instance, alpha helices and beta sheets that, in particular, the alpha helices can be also right-handed, and there are other turns, bends, and loops. Now, there are also other more complicated structures. So there is a complete hierarchy of structures. The next step are the supersecondary structures that are consisting in two or three consecutive secondary structures that form what is called a supersecondary structure or motif. So here I'm giving some example, for instance, of two antiparallel alpha helices joined together by another secondary structure, which is called a hairpin band. So, and there are many supersecondary structure that can be recognized using, for instance, X-ray crystallography. And actually, for this, one needs to crystallize the proteins, and then to irradiate them, illuminate them with X-rays, and then from the shape of the scattered light, the scattered X-rays, one should reconstruct the structure of the crystal, which is not entirely an entirely trivial task. So it is a very complicated task. And now let me say that there are also more and more complicated structure. One of them is the so-called domain folds, and these are sequences of a few supersecondary structures together with connected together with secondary structures, and all folds and supersecondary structures are very rigid, so they don't change. In principle, as I said, the protein is subjected to thermodynamical fluctuations, but even if thermodynamical fluctuations can be very strong, up to 70 degrees, the fold, so the geometrical structure of this part of the protein is always stable. After about a temperature of 70 degrees Celsius, the thermal fluctuation starts to be very strong, and the folds are destroyed, and the protein from the native folded states passes to a state which is unfolded. So a fold which appears relatively often in proteins is, for example, the Roseman fold, consisting of two beta alpha beta units. In which here this beta and alpha and beta are connected together by a loop, which is also a secondary structure. And there are others folds that I will not discuss here due to the lack of time. Now, the secondary structures and, of course, folds and the supersecondary structures are connected together in the so-called polypeptide chain, which is actually the protein, and which is called a tertiary structure. The chain, as I said, in the native state is folded. This means that it has a characteristic space conformation, which is held together by weak chemical bonds like hydrogen bonds. I mentioned them before. Also, motifs and folds have characteristic geometrical structures, which are, as I said, very rigid. And logically, motifs and folds can be seen as a subset of tertiary structures. Now, there are also proteins that consists of many chains connected together, and the number of chains is limited. So the chains are the tertiary structures, and one can put together, at least in nature, 1, 2, and 4 structures. For some reason, the number 3 was not chosen by nature. And the chains composing a multi-chain protein. So if you take the structures composing the quaternary structures, are not functional by themselves, so they are not working as enzymes, but actually they closely resemble functional one-chain proteins. And these quaternary structures are much more complicated, and they look to be the results of a later stage of protein evolution. Now, let's pass to the last part of this lecture, and about the astrobiological meaning of proteins. And here, Hervé has lectured about the down oh, the the top-down about the down-top approach. Here we will do the converse of the top-down approach. And so the idea is that we start from the modern organisms, and then we can look inside the biomolecules, remnants of the ancient biopolymers, as they was even when life started to exist on Earth. Of course, this is possible if the following two conditions are fulfilled. One is that the genetic code of the first life systems should based on one of the biopolymers, so DNA, RNA, or proteins, which are at the basis of present organisms. Otherwise, if there is no connections with the first life systems, so it will be hard to proceed in that way along the top-down approach. And the other condition is that the remnants of the genetic code of the first organisms should still be present and observable in the genetic code of modern organisms, or of course of fossils, if the genetic code of such ancient animals or organisms can be reconstructed. And now, let's see, I do very like this transparency, which is showing 5 sequences of 27 randomly typed letters. And as you see in blue, it is written, are marked the letters which are equal at the same place. For instance, at the second place, we have everywhere an L. At the third place, we have everywhere a D, and so on. And the probability that by chance, if, for instance, some monkey had typewritten such 5 sequences at random, these blue letters are exactly at those positions are very, very low, because it is 1 over 429,500 billions of billions. So the probability that exactly such letters, such blue letters are there is very, very low. And actually, these are not letters, but they are sequences of amino acids of a protein called the phosphoglycerate kinase, or PGK, which is at the basis of a very fundamental process taking place in virtually all living organisms, which is the conversion of sugar to alcohol, which is important just to get the energy. And so this kind of proteins is common in a lot of organisms. It is not important if such organisms are very simple, like the colibacillus, so the Escherichia coli, or complicated like human. But so why these blue letters are there exactly at the right position, always at the same place, even if the organisms are so different? The probability that this is true, that this happens, is very, very low. So it may be a result of convergent evolution, but as I said, the amino acids are highly interchangeable. So one can change a point like an amino acid inside a sequence, and this has happened because you see the red letters are mistakes that during the time have occurred. These are point-like mutations in which one amino acid has been changed, and the whole molecule will always fulfill its role of an enzyme. Sometimes it can occur an accident that for some reason one changes some critical amino acid at some point, and then the protein starts to get crazy and cause diseases to the organism that has such proteins. So it is certainly not a result of convergent evolution, but just all these mistakes looks like a case of divergent evolution, that during the time simply organisms underwent some mutation. And we can see the emergence here of a phylogenetic tree. So for instance, let's compare the sequence of this protein PGK for a horse and for a human. There are just two mistakes. And on the other side, human and horses are separated together by 80 millions of years. And if you go to, for instance, compare the code of a fruit fly and a horse, then you see that there are many mistakes. But this is not a wonder, because the appearance of fruit flies and horses is distant about in time, about 500 millions of years. So at the end, we can form this kind of diagram which relates mistakes and time. So fruit flies appeared before, and before horses and humans, and for this reason, due to mutations inside the proteins, occurred mistakes. But there is no doubt that they had some common descendant. So there are other mechanisms for which one could try to understand these astonishing similarities between the proteins of such different organisms. But for instance, horizontal gene transfer. But still, if one explores very well how these mechanisms of horizontal gene transfer work, it is very unlikely that they could explain these astonishing similarities in the sequences of PGK of such different organisms. And okay, this is a remark I will skip. And so let us rather concentrate in these relations of proteins with evolution. So we can imagine that, so we can consider that there are about 100,000 different proteins which have been expressed which can be found in eukaryotic systems. But inside all this large number of different proteins, there are much fewer different structural motifs and folds. And this fact, as I've seen before, is certainly related to the evolutions. And actually motifs, so this supersecondary structure, look like Lego bricks used in the construction of proteins. And folds are supposed to fold independently as separate units inside the protein, but it is very difficult to show it. And in some way, it is clear that the increasing degree of complexity of the various structures inside proteins may be connected with the increasing complexity of organisms during evolution. And in fact, so life, as we know, it is based on biopolymers. So the genetic code of each individual organism is stored in its DNA. And the information on the genetic code of life forms may be obtained from the sequences of DNA, RNA, and proteins of different organisms. So if you wanted to search for remnants of a common ancestor in the present DNA, it is natural to search inside the DNA and RNA or proteins because they are equivalent, because the information is the same. and to look if such remnants can be detected. So what we can look, so where should we search? Important is, for instance, the proteins that are working as enzymes in essential processes of life, because we can expect that such proteins was present from almost from the beginning, or even if the life at the beginning was RNA based, it doesn't matter. Such information should have been passed afterwards to DNA and to proteins. And so there are a lot of such proteins, for instance, this PGK protein that I mentioned before, but then there are also a class of proteins that is called serine proteases, and many, many others. So a remark that in many cases mutations and the fact that different amino acids have the same chemical properties, so that they are interchangeable, led to considerable changes in the primary structure of proteins during evolution. But as we have seen using the methods of statistics, it is possible to check where there are the identical letters and to estimate the probability that this occurs. On the other side, sorry. Another point so here is, sorry. So another these molecules are also important. So these proteins that are working like enzymes are also very important because they, as I said, should be folded in some particular way in order to recognize the substrate molecules. And since they work in that way, that they recognize these molecules and act on them like enzymes, and making possible reactions with them, then this structure that they have should be conserved. So it doesn't matter if in the course of the time the primary structure, the sequence of amino acids changed. Important is that the structure is always the same. So, for instance, the abundance of the same motifs and folds in the proteins of different organisms is certainly related to the need of keeping intact the shape of a part of a protein which interacts with particular molecules. Now, so apparently there is a hierarchy or a hierarchy of structures, starting from quaternary structures, going down to the primary structure, that it is for sure connected with evolution. For instance, let me mention the Rosman hypothesis that is also reflecting the same fact, but at the level of the DNA. So in more advanced organisms, there is an exact correspondence between protein folds and given locations in the genes called exons. So the DNA is a succession of sites which are called exons and introns. Introns don't act very much. They seem to be there just to correct the possible mistakes in the replication of DNA. But exons are those that are used to code proteins, and they are coding the folds. So this suggests that not only proteins have substructures that are related to evolution, but also inside the DNA, there are structures that are also related to probably more primitive genes, which afterwards, after some time, join together by some process of gene fusion in order to give rise to more and more complex organisms. So, indeed, this hypothesis is very reasonable because it is very likely that the first organisms had a very simple DNA which encoded very basic proteins. And only during the evolution, DNA and proteins have become more and more complex. Now, the problem is that folds are almost certainly the remnants of fold proteins. But what are the simplest possible structure that folds and that can be interpreted as the first protein, as the first proteins? I would like here to mention the so-called Trifonov hypothesis, which is due to Edward Trifonov, which is based on physical and chemical considerations. So we have seen that the proteins have a C-terminus and an N-terminus. So these are the ends of the protein. And the C-terminus is a little bit dangerous because in that part of the protein, one can add further amino acids. So if the proteins are very, very simple, then in some way this C-terminus must be shielded. And the idea is that to protect this C-terminus, a very, very fruitful structure is that of a loop, because then the C-terminus and the N-terminus will be very, very near, preventing the contact with other dangerous molecules like other amino acids. And if you check the rigidity of the molecules, then one can see that the loops are formed if the length of the protein is about 25-30 amino acids, which means, if I'm not wrong, that the corresponding DNA or the corresponding some logic jump RNA is about 160 nitrogen bases. And up to now, this hypothesis has been checked by numerical simulations of crystallized proteins that show loops that loops of that size are a major structural mode. So they are appearing very, very frequently. And on the other side, Trifonov made also statistical research inside the protein databases, like, for instance, the PDB database. And he has found that the following sequences, so there are 7 sequences, sorry, of amino acids, which are containing from 25 to 30 amino acids, are appearing inside a lot of proteins in different organisms very, very frequently. So the idea is also to test this hypothesis. This is what I wanted to do, and in fact, for this reason, I bought equipment, calorimetric equipment to do that. Equipment that is able to detect all the exchanges of heat when the polymer folds. So in that way, it will be possible, I hope, to see that if to check if really proteins have structures very, very small, like those substructures of Trifonov that can fold independently. If not, of course, there are always limits to the technology. Of course, I could always use calorimeters for other projects, for instance, biomimetics, for instance, to study how the wine is tasting. So let me finish giving you a very, very basic bibliography. I'm very sorry that I didn't put important references, like for instance the articles of Trifonov. And finally, let me thank you for your attention. Thank you.
Thank you very much, Franco. I have a couple of questions, or certainly one which is important, I think. You briefly discussed and made a couple of examples to a particular enzyme, making comparisons between the sequence of amino acids between horses and humans and horses and Drosophila. Now, while you generate some numbers like 80 million years, I'm just wondering whether looking at protein or differences in proteins between species is the way to go, because clearly if you look at the genetic code, it's where you have the third base pretty much redundant for the majority of the amino acids. I'm wondering if you do the same sort of analysis at the genetic level, whether you come up with similar numbers.
Sorry. Mm-hmm. So you mean that so?
I'm just saying that you're doing an analysis of a bit at the amino acid level.
Yes.
But if you do the same analysis on the gene, on the actual DNA level, whether you come up with similar trees and distinctions in phylogenetic tree.
Well, I think since proteins are expressed by RNA, there should be some correspondence. I don't think that, and there are no analogies between these coincidences in interposition of amino acids and the position of the nitrogen bases in the DNA. Actually, the alphabet is different because the DNA has just 4 letters. Here we have 20 letters. So maybe some of the nitrogen bases can be interchanged. So maybe this is why, as you mentioned, that there is some differences. But at the level of proteins, we see. So it has been shown that not only this PGK, but also other proteins like dehydrogenases and many others have this astonishing coincidences between the position of the amino acid residues on the chain.
Okay.
So,
thanks very much.
Thank you for the question.
I'll pass the question mic to Alessandra, University of Napoli. Do you have any questions there?
There is one question.
Okay.
I know that the RNA appears on the earth as one of the first enzymes millions of years ago. And why was the RNA replaced by enzymes, by proteins composed by 20 amino acids? And so my question was, why was RNA replaced by proteins, and so then it became an informational molecule instead of being an enzyme?
Mm-hmm. Well, but that's a good question. It's a very difficult one. So why we should switch from an alphabet of 4 letters to an alphabet of 20 letters. So I guess that RNA, of course, at the beginning was acting as enzyme and was acting also. There is a large consensus about that. It was acting also as a memory, a code, just to memorize all the functionality of the organism. So why one should pass? Why it was very convenient to pass to proteins? Well, this is a very difficult question. I guess in some sense the talk of Hervé has answered the question in the sense that we have seen that amino acids are very easy to be done. Instead, the sugars that are at the basis of the DNA and of the RNA are much less, are much more degradable. So there is this is really a problem, because if we have the sugars that are at the basis of DNA and the RNA, and you keep them at the temperature of 70 degrees, they degrade in a while. So even if you have some room temperature, they can degrade in a few hours, if I'm not wrong. So in the case of organisms, where so if you have a much more complex organism, that it is living also in conditions that are not very favorable, because the temperature can be higher or lower. So it is much better to ensure how this organism will function using molecules that are much more stable. So I guess that for this reason, in the course of the evolution, proteins appeared, because proteins have this peptide backbone, and this is very strong. In the same way, I think in the seminar of Hervé was appearing this alternative of DNA, which is based on also on peptide bonds, but of course always on sugars. But the idea, so my answer is that during the evolution, it has been important for organisms to have some way to keep the functionality even in conditions that were not really favorable. So instead of sugars, one used proteins that are very much more stable. And on the other side, as we have seen, it is full of amino acids, not only on the earth now, but in the universe. So I hope that I answered. But this was a very good but difficult question.
Yes, it's satisfying answer, conceptually. Thank you very much.
Thank you. So some sense.
Are there any other questions, Alessandra, from your students?
No more questions. Thank you.
Wonderful. Thank you very much.
Yes, I have. Thank you very much. My question would be that right now in biology, we think, or we know that DNA basically does the design, and proteins basically, or peptides basically do the building, or be the building. And maybe the key to the responsibility of the building is the folding, or the folding procedure, that these proteins can have lots of levels, like primary or secondary structures. Are there any other molecules that can be synthesized in vitro, or any other artificial way that can have these folding probabilities? Or are there any molecules that can act similarly to proteins? This would be my question. Thank you.
Oh, that's also a good question. So, so of course, if there are other molecules that can fold in such a way, I guess any molecule that can support this hydrophilic, hydrophobic, and van der Waals bonding. But actually, I'm not so expert to say. So I've seen a lot of seminars actually, but if there are alternatives, of course there are proteinoids, so there are prions, but other kinds of almost proteins. But if the other molecules can fold, I don't actually know. But the next time I will know.
Thank you very much.
Thank you very much.